This video is part of the series on plants, primary structure and growth, and relates to Chapter 35 in Campbell Biology. In this video, I'm going to talk about the three primary tissue groups. The three primary tissues in plants are dermal tissue, ground tissue, and vascular tissue. In the diagram on the right, we can see that they've been color-coded. Dermal tissue surrounds and envelops the outside of the body and has been color-coded this blue cyan color. Ground tissue is color-coded yellow and fills the middle of the body, and vascular tissue groups into strands or bundles, is continuous throughout the entire body, and is color-coded purple in this diagram. The diagram also shows us the typical anatomy and arrangement of these tissues in the three different primary organs. At the top here, we have a cross-section through a diagrammatic leaf. We can see dermal tissue on the outside, bundles of vascular tissue, and then filling the middle between is the ground tissue, shown in yellow. Next down, we have a cross-section through a typical eudicot stem. Again, the outside we see dermal tissue. The bundles in purple show us bundles of vascular tissue, and the yellow, surrounding and throughout the middle, represents the ground tissue. At the bottom here, we have the cross-section through a root organ. The outside is labeled dermal tissue, the center bundle of vascular tissue, and then in yellow, the ground tissue. These three tissues, dermal, ground, and vascular, arise from the action of totipotent stem cells called initials, undergoing mitotic divisions to produce new cells from the meristem tissues located at specific points in the plant body. Throughout this video, notice how the form or anatomy of each tissue relates to its primary functions. The first tissue I want to talk about is dermal tissue. Dermal tissue is an external layer of tightly packed cells. It functions in protection, including protection against pathogens, herbivores, to prevent desiccation of the body, and to prevent entry of too much ultraviolet radiation, and many other substances as well. It also functions to regulate what substances enter or leave the body, including carbon dioxide, which is important for photosynthesis, oxygen, which is important for cellular respiration, and the uptake of water from the root system and the loss of water from the shoot system. In form, we see that dermal tissues have cells that are tightly packed together, and these cells also have the ability to produce waxes in the living cytoplasm of the cell. The waxes, as they're produced, are shipped to the edge of the cell and released from the cell membrane so that they build up in the cell wall, in and on the wall. Because the waxes are produced inside the cell and brought to the edges of the cell can control what surface it ships those waxes to. And so we'll see that in dermal tissue, there's often a polarity from the external surface of the cell, which may have a buildup of waxes and a protective layer, to the internal side of the dermal tissue, which faces the internal tissues, and we don't see the waxes there. There are two main types of dermal tissue in plants. Epidermis is what we call dermal tissue that covers the primary growth structures. These are the young, typically green structures of the plant if we're in the shoot system, or the roots in the root system. And then there's periderm. Periderm is a type of dermal tissue that develops after the plant has begun secondary growth. Remember, that's growth that increases the organ's width, and it often shows up in more woody parts of the plant, including both woody stems and older woody roots. The epidermis is a single cell layer, whereas the periderm can be many cells thick. In this picture here on the upper right-hand side, this is the epidermis here, and these are two very specialized epidermal cells called guard cells that I'll be talking about in a minute. And in this lower picture, we can see the periderm here is this thicker layer of several cells. So let's look at the epidermal tissue. Remember, this covers the primary body of the plant, organs that have undergone only primary growth. In addition to the epidermal cells, which form a tightly packed single layer here, this is a leaf. This is also a leaf here. These epidermal cells are parenchyma-type cells. They're alive at maturity. We can also see that there's some very specialized epidermal cells, such as these hairs. These are called trichomes. And these cells in here, there's actually a pair of cells here called the guard cells. These are also specialized epidermal cells. Trichomes can be unicellular or multicellular. They develop from specific cells within the epidermis, which grow, elongate, and eventually can divide mitotically to produce multicellular hair structures. Sometimes these hair structures also have glands to release substances that help prevent herbivory or fight off other potential attacks from animal predators. Here's some more pictures of the epidermis. So if we look down here, here's our epidermal cells. This shows a really thick layer of waxes that have built up to the exterior surface of these cells. These are also living epidermal cells here with thick exterior wax. That wax that builds up to the outside of the epidermis is called the cuticle. And it plays a very important role in blocking entry of pathogens and preventing having too much loss of water from the shoot system. There's also an important pair of cells right here. Here's one, and here's the other. These two cells together are the guard cells that together surround a stomatal pore, a singular being called the stoma, a stoma pore. The two guard cells, if we were to look at them from a bird's eye view, would appear either kidney bean or sausage shaped. So here's one on this side, and here's the other on this side. And together they surround to their inner edges a pore which allows substances in the vapor phase, like water vapor, or gases such as carbon dioxide and oxygen, to move into the organ or move out into the external environment. These pair of guard cells have the ability to control how wide that stoma pore is or how narrow it is, either increasing or decreasing the rate of gas exchange between the organ and the external environment. We'll learn more about that in the section on transport, but for now I'll just tell you that it has to do with the amount of water pressure inside the cells and the way that they develop with certain anatomy in their cell walls. Another specialized epidermal cell are the epidermal cells in the root system that form root hairs. Unlike trichomes, which are often multicellular, root hairs are tubular extensions of a single cell. They do not mitotically divide, and they remain attached and part of a single epidermal cell. So this here is this tubular extension off of the epidermis cell in the root system. Just like any other cell, it has a cell membrane, which is continuous through the root hair and out and across the rest of the epidermal cell. And it has a thin primary wall that covers that cell membrane because it's a parenchyma type cell. These tubular projections hugely increase the amount of surface area of the root, thereby really increasing how much area across which water can be taken up and minerals. They're therefore an amazing adaptation to help plants obtain the resources that they need from the soil. The next tissue I want to talk about is vascular tissue. Vascular tissue is packed into bundles called veins or vascular bundles, which are either located in strands in some organs or as a central cylinder in organs such as roots. The vascular system, remember, is continuous throughout the entire plant body. So in any organ that we were to look in, we would be able to find vascular tissues. The main function of vascular tissue is long-distance transport and also structural support. There are two main vascular tissues. They are known as the xylem tissue, spelled X-Y-L-E-M tissue, and the phloem tissue, P-H-L-O-E-M tissue. On the left-hand side, we can see a tissue that's been cut in longitudinal section, and staining pink, we can see an area of xylem tissue here. And then just to the right of that, not staining very heavily at all, is an area of phloem tissue. There's also some other cell types in there that you should recognize, such as sclerids, fibers, and generalized parenchyma. On the lower right-hand side of this slide, you can see a cross-section through a stem organ. I'm pointing right now to the epidermis, and just under the epidermis is an area of parenchyma cells providing some support. But what I really want to call your attention to is this vascular bundle here, which I'm circling. This vascular bundle consists of three parts. The outer part here is a stack of fiber cells. Interior to those fiber cells in this area here is the phloem tissue. And interior to that is the xylem tissue. So let's talk about these two tissues in turn. We'll begin with the xylem tissue. The xylem tissue's main function is structural support and long-distance transport of water and minerals from the root system, where they're obtained, to the shoot system. There are four cell types that we commonly find in xylem tissue. These are tracheids, vessel elements, which are mostly found in the flowering plants called the angiosperms, fibers, and some generalized parenchyma type cells. These first three, tracheids, vessel elements, and fibers, are all types of sclerenchyma cells, meaning that they are dead at maturity and they have a thick secondary wall containing that highly branched, rigid, and strong compound known as lignin. So a little bit more about these two very specialized sclerenchyma type cells called tracheids and vessel elements. The tracheids and vessel elements are the cells through which the water and minerals actually flow. They provide the conduits for the movement. Tracheids are found in nearly all vascular plants. They are a long, thin cell with a tapered end and lignified secondary walls. And by lignified, I mean containing that carbohydrate that we just talked about, called lignin. Water moves from cell to cell inside the tracheids, which are hollow and dead at maturity, and then travel between tracheids through special points in the wall, which have become very thin and are called pits. The pits of one tracheid line up with the pits of the adjacent tracheid so that the water can easily move from one tracheid to the next. Vessel elements also provide a conduit for flow of water and minerals. The cells and structure are wider and shorter, and they stack one right on top of the other to form long conduits. The end walls, where one vessel element stacks below and adjacent to the next vessel element, degrade in large part to form what's called a perforation plate. On the upper right-hand side here, you can see a perforation plate, and you can see how much of the wall structure of the cells degraded. Water can flow freely, therefore, through one vessel element and up into the next vessel element, forming long tubes. Vessel elements do not form tubes that go all the way with these open end walls from the roots to the shoot, but rather stack to form several vessel element long tubes, and then they have a tapered end, eventually, like a tracheid, forcing water to eventually move through pit pores, which they also have in their sidewalls. These pit pores are just like those in the tracheids. They are areas where the wall material has become thinned and where the pit of one vessel element aligns with the pit of the next vessel element. In most species, vessel elements are interspersed among tracheids. In this image down here, in the lower right-hand side, you can see these narrower tracheids and then these larger vessels. The water can also flow from vessels into tracheids because they both have pits in their sidewalls. Phloem tissue is the other major vascular tissue, and its main function is to transport sugars and various metabolites from what's called the source, which is the area of the body in which they were produced, to what's called the sink, which is an area of the body where they're needed, such as a rapidly growing tissue or a storage organ, which is busily trying to store up sugars for maybe an overwinter storage. Again, we see 4 main cell types comprise the phloem tissue. 2 specialized cell types and 2 which you've already met. The 4 cell types are sieve tube elements, also sometimes called sieve tube members, companion cells, generalized parenchyma cells, and fibers. These first 3, sieve tube elements, companion cells, and parenchyma, are all parenchyma-type cells. Just the first 2 have become highly specialized for their functions. So let's talk about sieve tube elements, both their structure and how that structure allows their function. Sieve tube elements are parenchyma cells, which means that at maturity, they are still a living cell. They have a thin primary wall. They are long and narrow, and they stack end to end, much like we just saw with the vessel elements, except the vessel elements were sclerenchymas, and these are parenchymas. But they also tend to have some degradation of their end walls between where one sieve tube element butts up against the next sieve tube element, allowing them to form long chains that form conduits through which long-distance flow can occur. The name of the end walls, which degrade and have large holes in them, has a different name though. In sieve tube elements, the end wall that's degraded to have holes in it is called a sieve plate. Another really interesting thing about sieve tube elements is that in order to allow for efficient flow through a cell that is still living and is therefore not hollow but is actually still filled with cytoplasm, a lot of what we consider the main important organelles inside these cells during maturation slowly degrade and break down, and their nutrients are reabsorbed. So that at final maturity, a sieve tube element typically lacks a distinct vacuole, has very minimal cytoskeletal elements, lacks a nucleus, and lacks ribosomes. In this longitudinal section here, shown with a light microscopy, we can see several sieve tube elements that are immature over here and are still small. And then we can see some mature sieve tube elements here. And this is where they're stacking together and where we would find the sieve plate if we were to look at it carefully. We can also see at the edge here a little bit of developing xylem tissue. This has some nice pictures of the sieve plates on it. So down here on the lower right-hand side, we can see a scanning electron micrograph of 2 sieve tube elements where they abut each other. The end walls degrade to form holes, and we get the sieve plate. In diagrammatic form, it would look like this. Here's a sieve tube element. Here's another sieve tube element, and where their walls touch here, we get the sieve plate. Now you might be wondering how on earth a cell can stay alive and not have something as essential as a nucleus. And the answer is, for every single sieve tube element cell, there is an adjacent cell called the companion cell. And while the job of a sieve tube element is to allow for efficient flux of fluids, the job of the adjacent companion cell is to play chaperone to the sieve tube element. The companion cell, since its main job is to chaperone the sieve tube element, as well as regulating itself, must be located directly and adjacent to every sieve tube element. The companion cells themselves are non-conducting, meaning they are not part of conducting fluids through the vascular tissue. They simply exist to regulate both themselves and the sieve tube element next to them. To facilitate this regulation, they have a dense cytoplasm with many organelles, and they're connected to the adjacent sieve tube element by a high density of plasmodesmata. Up here, towards the upper left side of this picture, we can see a cross-section shown in transition electron microscopy of a sieve tube element on the left side and a companion cell on the right side. And you can even see these little lines here are where this slice that was made through the tissue cut directly through some of the plasmodesmata that are numerous connecting these two cells. The final tissue I want to talk briefly about is ground tissue. Ground tissue is sometimes called soft tissue because it's composed mostly of parenchyma. There's also some collenchyma, but there aren't any sclerenchyma. And because most of it is parenchyma, and parenchyma has a thin primary wall, ground tissue tends to be fairly soft in nature. Some examples of ground tissue include pith tissue, which exists in stem organs interior to the vascular bundles, cortex tissue, which exists surrounding or external to vascular tissue, and mesophyll tissue, which exists in the middle of leaves. This diagram over here shows us two different organs in cross-section. Up here we have the stem in cross-section, and down here we have a root organ in cross-section, and it's color-coded to show us how the ground tissue is the tissue in the middle, between the vascular tissue and on the outside the dermal tissue. Again, this is a cross-section through a root organ. Up here we have the cross-section through the stem organ. Again, color-coded is the dermal tissue on the outside. The purple represents the vascular bundles. And then the yellow, again, represents the ground tissue. So the pith would be here in the center, and the cortex would be over here. Ground tissue, because it is mostly comprised of parenchyma-type cells, functions mostly for a variety of metabolic purposes, as well as a little bit of flexible support for growing bodies because it contains collenchyma, and of course some short distance cell-to-cell transport within the tissue. Some examples include photosynthesis, which is the primary function of the mesophyll tissue, which is the ground tissue in leaves. Storage, so for example, the flesh of a fruit or the soft internal tissues of a potato are storage for sugars and starch respectively. Support, like what we see here in this cross-section down here, where the outer part of the ground tissue is made of collenchyma, and that collenchyma, remember, has an irregularly thickened primary wall, providing flexible support for growing organs. And then also short distance cell-to-cell transport across the tissue. So if we come back over here to look at this diagram of a root and cross-section, if substances are being taken up across the dermal tissue, they then need to be transported across the ground tissue to get to the vascular tissue in the center, from where they can then undergo efficient long-distance transport. So in this lecture, we talked about the three primary tissue types in plants. We talked about dermal tissue, ground tissue, and vascular tissue. And we looked at how tissues are groups of specialized cells, and we looked at several of those specialized cells in some detail, that together form an anatomy that is adapted to perform specialized functions or physiology.